Welcome to Protein Translation, the third video in the genetics video series by Mr. Caputi. Before you watch this video, you should have read and answered the What is DNA question and sheet. You should have also watched the DNA Basics video. You should have taken a quiz on DNA Basics. You should have watched the mRNA transcription video. You should have read and answered the questions to transcription and translation overview. And you should have just completed taking the quiz on transcription. Today with this video, you should follow along on the handout called Protein Translation. Feel free to take notes on the paper or take notes in your journal. You will definitely need the table that is listed on that paper in order to follow along with us for today. Now let's just do a quick review. Remember, this molecule is called DNA. It contains all of the information that makes you, you. All right, the specific letter code of DNA allows all of the proteins in your body to be made. Remember a couple of other things from previous lessons is that the process of turning DNA into RNA occurs in the nucleus. It is a process that is called transcription. Whereas turning that mRNA into an amino acid chain happens at the ribosome, if you remember our friend the ribosome from our cell unit, and is a process known as translation. Now today we're going to be focusing significantly on the translation side of this process. Hopefully by now you can already go through the transcription pretty well. So to do a quick review of where we stand, remember that our DNA is trapped inside of our nucleus. Now the reason for this is a protective reason. Remember, the letter code of DNA can change pretty easily if it's damaged, and so the cell does not want that DNA, pretty much all of the information that makes you, you, to escape the nucleus. All right, if it were out of the nucleus, it would be very, very easy to damage, and as soon as you change that letter sequence, you can change a lot about your body. So now in order to solve this problem of the fact that the DNA is trapped inside but needs to essentially get to the ribosome, because remember the ribosome is in charge of making proteins, the way the cell works around that is by making a copy of a small section of the DNA. All right, now remember that process is called transcription, and what you're making there is mRNA. That's what was covered in the last video. And to do a quick review, if this were the DNA sequence that were trapped inside, you should be able to transcribe the sequence into an mRNA sequence. All right, we're going to take about 30 seconds right now in order to do that. Pause the video and transcribe this sequence on your own. So just press the pause button and then transcribe the sequence. Once you're done, press the play button and we'll continue. All right, now that you've pressed play again, what we're going to do is go through and make sure that you are good with your transcription. So if this were the sequence here, all right, the A in DNA would become a uracil, then you'd have an adenine and a cytosine, then a guanine, then an adenine and a uracil, then a cytosine and a cytosine and a guanine. All right, remember the tough thing here is that these U's occur only in mRNA and they don't exist in DNA. All right, remember uracil is only in mRNA, it's not in DNA because there's no thymine in mRNA. All right, so now we've already gone through this before, but if you'd like to practice one more time, practice transcribing this sequence. Press pause, and when you're ready, after you've done your transcription, press play again and we'll go through the answer. Now that you've pressed play, let's see how you did. Hopefully this is the sequence that you came up with. All right, if not, I suggest that you go back to your transcription video because you might not be ready yet to move on to translation. If you did get it right, you're ready. All right, so now let's get started with translation. Now translation begins after transcription has already occurred. All right, so we have this fun little mRNA sequence right here that's just been copied. And now since it's mRNA and it's not DNA, it's free to leave the cell. So what happens is, or free to leave the nucleus, I'm sorry. It leaves the nucleus and moves over to the ribosome. Now remember, the ribosome is the organelle that's responsible for making proteins, and this is exactly where the information needs to get. So once the mRNA binds to the ribosome, now the ribosome can do its job. All right, here's a little zoomed in version of that. Essentially what the ribosome is going to do is it's going to read this sequence, but it's going to read it in a very specific way. It's going to read it 3 letters at a time. All right, the first letters it's going to read are the U, the A, and the C. So what's going to happen here is that this section of mRNA is actually going to get fed in and go U, A, C, and get read by the ribosome. All right, now the ribosome, its abilities allow it to read those letters and figure out which amino acid that codes for. Now, amino acids are important because they're essentially the building blocks of a protein. All right, think of a chain and all the individual links in a chain. Each amino acid is like a single link in that chain. Now, which amino acids go into that chain really control what type of protein is formed. All right, if you have a lot of certain types of amino acids, it could do one thing. If you have different amino acids in a different order, it'll do something completely different. An example we used before was the difference between brown eyes and blue eyes. There's only 2 amino acids that are different between the brown eye protein and the blue eye protein. Now, the ribosome is able to read our first 3 letters, UAC, and pull out an amino acid from it. In order for us to be able to do this, we're not quite as talented at the ribosome at this, and so we're going to use a little bit of help. The help that we're going to use is in this table right here. This is called a translation table. All right, so this translation table is going to be responsible for showing us which of the 26 different amino acids the 3-letter codon, so those codons, remember, are the 3 letters in a row that code for an amino acid, which amino acid that codon controls. Now, how does this work? Well, it looks pretty complicated, but in reality, it's pretty simple. So we're going to get started with the example of the codon CAU. So that's part of an mRNA sequence, just any mRNA sequence. It could be CAU. We're going to start with the C, because that's the first letter in here. All right, so what you need to do is you need to take your first letter of your 3-letter codon and compare it to the first column here. All right. And so once you find it's not the G, it's not the A, it's actually the C right here. So what this tells you is that the amino acid that we're looking for has to be somewhere in the C row. Now, it could be any of these, but we know it has to be one of these. All right, so now that significantly narrowed down our options from all 26, because now all we're left with are, it could be leucine, or it could be proline, or it could be histidine, or it could be glutamine, or it could be arginine, or it could be arginine. All right, incidentally, that's part of my favorite joke. What is a pirate's favorite amino acid? That's right, it's arginine. I love that one. Okay, so back to the science. After we've taken a look at the C amino, or the C first letter of the codon, we then move on to the A, the second letter in the codon. Now, as you can probably guess already, what we're going to do is we're going to take this A and we're going to actually compare it to the top, right here, all right, the second base in the codon. So we know it has to be somewhere in the C row. It also has to be somewhere in the A column. So it has to be one of these in here. Now, the nice thing, since we know it's already in the C row and it has to be in the A column, the only place that we should be looking is right where they intersect. So it's got to be one of these 4. It's either a histidine or a glutamine. All right, now the last step, some of you might already be jumping there, is realizing, what do we use the 3rd letter for? Well, the 3rd letter determines which one of these 4 rows is our letter. In this case, our 3rd letter is a U. So we're looking in this row, which means the amino acid that this codes for is histidine. Bum, ba, bum. All right, so now let's use this table a little bit more and get some practice on our own. As you can see, this new mRNA sequence at the bottom is the same mRNA sequence that we were using in our original example. So what I want you to do is go through on your own and try to practice to figure out which amino acids these would code for. Now we're going to do the first one together, and then I want you to do the second 2 codons on your own. All right, so your first job here is to split it into your triplet codons, so 3 letters. So UAC is going to be our first one, then GAU, and the last one is going to be CCG. All right, so remember what we do. We start with our first letter in the codon, which is our U. So that means it's got to be somewhere in the U row. somewhere in here. Our next letter is A, which means it has to be somewhere in the A column. All right, so thankfully that narrows it down to either tyrosine, tyrosine, stop, or stop. And we'll talk more about what stop means in a second. And last but not least, we need to take a look at our last letter, which in this case is C. Figure out which of our 4 lines is C. This one is C, which means the one we're looking for is tyrosine. All right, what I want you to do right now is press pause on the video and take care of the second 2 codons. So I want you to translate GAU and figure out which amino acid that is, and then translate CCG and figure out which amino acid that is. Press pause now. All right, so hopefully what you're able to find is that the next 2 you were looking for is aspartate and then proline. All right, if you successfully did that, that means you are on a roll. If not, I would suggest rewinding a couple of minutes in the video and going back and practicing some more. All right, so how would this actually look in a ribosome? Well, if we go back to our friend the ribosome, what it's doing is it's reading UAC, and what it does, it says, hey, UAC, I know what that one is. That one, as we just discovered, is tyrosine. So what it does is it searches for a tyrosine amino acid and brings it over to the ribosome and clicks it into place. Then what it does, it reads our next triplet. GAU, it says GAU, I know what that one is. That one's aspartate. So what it does is it brings an aspartate over to the ribosome, connects it to the tyrosine, and then both of those move over. And now the chain is a little bit longer than it was before. Now it's a tyrosine and an aspartate. And this will continue. CCG, ding. CCG, that's a proline. So what it does, it brings over a proline, attaches it to the end. The whole chain moves over. And now we have a chain that's 3 amino acids long. Now this will continue and continue until you get hundreds of amino acids in a row. All right, essentially this would really continue until you finally hit what's called a stop codon. So this brings us to the other 2 kind of unique, the unique amino acids that you might find. All right, there's 2 really unique looking amino acids on our table. All right, what you'll notice is that from our chains, you'll see that this one, methionine right here, says start next to it. And then there's also a few of them that say stop. All right, almost all proteins begin with the methionine amino acid. It's kind of a signal to the mRNA that it needs to begin work. All right, so as soon as it reads an A, U, G, that says, oh, hey, look, we got a new protein. We're going to bring a methionine over, and it usually will be probably 8 methionines in a row on an average protein. Now, it'll keep attaching amino acid to the end, to the end, to the end, to the end, until finally it hits one of our 3 stop codons. As soon as a ribosome reads one of the stop codons, it releases the entire chain. At that point, the whole chain actually leaves the ribosome and goes out to do its business. All right, it's either going to go into part of your eyeball to make an eye color. It's going to go into your stomach to digest some of your food. It has any number of functions of proteins. Now, protein functions are outlined a little bit more in the reading, and I really suggest that you take a look at that. It's on the back page. It's essentially the last 3 paragraphs with a list of what some of the other amino acids do. I'm sorry, what do the other proteins do? All right. This is a little bit more practice, so this is the transcription that we had done at the start of class. One more time on your own, just to make sure that you have it together. What I'd like you to do is pause the video and translate the red mRNA sequence that you had made from before. So press pause now. All right, now hopefully you've been able to translate this sequence, and your answer should be serine, threonine, histidine, leucine, cysteine, glycine, alanine, and it should end with a stop. If you've reached that, congratulations. You can translate mRNA sequences. And you are ready to practice on your own. If you weren't there yet, either rewind the video or reread the reading on your own. What you should now do is first, number 8, finish the protein translation questions. So there's 3 transcription review questions and then 3 translation questions. After that, read and answer The Danger of Giving Bad Directions. It's another reading about mutations and how they can affect proteins. And when you're ready, take the translation quiz. Good luck, and I'll see you next video.